Carlos was educated at Miami and Pennsylvania, and he's done his postdoctoral positions at University of Texas at Austin and at Harvard. And the last years he's been at Vanderbilt, and he's leading research group in computational biology. So he'll talk today about intracellular signaling processes and cell decisions in stochastic algorithms.
Thank you, Radek. So, again, I had a similar. I should have gone TBA instead of this title because I'll talk much less about intracellular signaling and more about cell decisions. Mostly because the stuff that I'll be talking about is very preliminary. So it'll be interesting what you guys will come up with. But I'll tell you a bit about our modeling framework, and I'll tell you a bit about the problems that we're interested in. This is just a brief outline. I'll start with a little bit of our biological motivation for this. I'm in a medical school, so we're surrounded by a lot of interesting problems that are very difficult to understand. I'll introduce a bit of our PySB modeling framework, which is a tool to model different biological programs using Python as a native language, which I think is pretty cool, and I'll be happy to talk about that more. And then I'll show you a little bit of our application for this. So as I said, I'm in a medical school, and particularly I'm in a cancer center. And so we do what we'd like to call cancer systems biology, and so we do a lot with drug treatments. And the way that this works usually is, the pharmaceutical companies have a molecular characterization where they have a compound and a target, and they come up with an inhibitor. In this case, I'll talk about erlotinib fairly regularly. And then once you have that inhibitor that is targeted to some molecule, you do in vitro proliferation assays. So you take your cells, you culture them in a little dish that looks like that, without the pretty lights, and then you see how your cells grow. And then if everything works really well, you go to the in vivo animal studies, and then eventually you move on to clinical trials, and hopefully after a few years, 5 years, 10 years, something like that, and if everything goes well, then you find a drug that works. But there are problems along this way. This pathway is very long. It takes about 10 years or so to go from a drug from molecular characterization to eventually clinical trials. And there's complications along the way, many complications along the way. And what I'm going to focus on today deals a lot with in vitro proliferation assays, where you have a drug that seems to work, but we know that there's I don't have to tell this to this crowd. There's a lot of noise in these systems. So how can we actually understand the systems better? And the traditional view is that these drugs work very well. People do this in vivo characterizations, in vivo characterizations, and they measure the efficacy of the drugs based on concentrations, and they see how efficient the drug is, and then they have all these different characterizations for how good this drug is. And they might go and look at potency, maximum efficiency, or whatever. And these metrics are static, and they give you a one snapshot, but they're not very good at characterizing the population behavior. And we actually just heard that the paper got accepted in Nature Methods, so we'll talk. The paper will deal a little with some of this stuff. But today, I'll tell you about what happens at the single cell level and the single cell population level. Small populations and how single cells respond differently. So our collaborators, Vito Quarenta and Peter Frick, did some experiments where they did this so-called clonal proliferation assay. You have a bunch of cancer cells, and you pull out one of them, and you plate them on a dish, and you plate them individually. And then you let them grow. And the idea is that once you let them grow, you watch the dynamics of growth in the presence of drugs. So you let them grow for a few generations up until you get about 10,000 cells also. And then you treat them with drug, and then you watch how they respond. And what you see invariably is that if you do the whole cell population, you'll see something where they kind of go through an adaptation period, and then eventually some of them will keep growing. Some of the subclones, I'm going to call them subclones, some of them will keep growing, and some of them will actually continue to die. And eventually, they will if they have a negative slope, they eventually die. If they have a positive slope, they continue to divide. And it seems that this mixture of things is what contributes to a tumor continuing to grow. So what we did with this is we characterized this, what we like to call the drug-induced proliferation rate. This is how the cells respond in drug as a population. And what we see is that these rates actually have a pretty normal distribution. So for example, DS9 cells, which are shown here, no, sorry, this is DS1, but they have a very similar dynamics. They have a very small proliferation rate. whereas the other ones here have a higher drug proliferation rate. So when we started when we got into this deal, the question was, well, what is it that's giving rise to this differential proliferation rates under drug, and how can we understand this? And in the traditional view, in the cancer field, is that you have the drug sensitive cells, and then you have the drug resistant cells, and there's a genetic component to that, and that's pretty much it. But this idea is borrowed from I guess some of the ideas that have been borrowed from work in prokaryotes or even smaller eukaryotes and things like that. They've seen that there's actually more than one. There's more states than this. There's actually the cells that are sensitive, and then there's some non-genetic tolerance that we like to call tolerant, and then somehow the cells could eventually become genetic resistors. So the question is, how does this sensitivity to these things arise? And there's been a lot of questions about this. And just to show this experimentally, this is an actual experiment where they took cell populations and they seeded them, and then they watched their this is log 2, so this is just population doublings over time after treatment with drug, and they see that some populations go up, some populations kind of stay steady. But again, the traditional explanation is, there's just an average there, but can we do better? So this is where we came in, and we decided to start looking at these things kind of from a different perspective. What we have done in my lab is develop a modeling framework that allows you to basically encode models and look at them in different contexts using different programming tools. So from my view, my background is condensed matter physics and computational massive computation. And from my view, the challenge here is how do we go from the math representation of the systems to the biology representation? And one of the problems is that by the time we write the math, we've kind of lost a lot of the description of the biology. So how do we actually do this? So with our PySB framework, what we focused on is looking at actions and what we call action-based or program-based modeling. And the idea is that when you talk to an experimentalist, and we've heard in different talks today, people will say, Bax translocates to the membrane, or Bax oligomerisis makes a pore, or one of this BCL2, MCL1, and BCL-XL inhibits BID, or BID activates Bax. And the question is, can we encode this as a program so that we can actually manage it and probe it better? So that was my idea. And so what we've done is we've created this tool where we can make models in Python, and this is the actual code for a very simple ligand binding. And what you do is you just make this ligand receptor thing. We declare some monomers, some parameters. Right here, these are initial conditions. And this is just a simple rule, but it could be functions. It could be the oligomerization function. It could be the catalysis function. It could be whatever you want. And once you have the model in a computational framework, you can simulate it with ODEs or whatever. And the way this works is you declare your model. You call some tool like BNGL to make the reaction network. You make an ODEs graph. This returns it to Python, which then makes an ODE or makes a stochastic model or a spatial model or whatever you want to do. And then you can solve it, and then you can do whatever analysis you want. So we did this so just to kind of hammer that message home. What we're doing is you guys are very good, or we are very good at writing these equations, and the tools like BioNetGen and Kappa are very good at connecting the protein-protein interactions to those rules. And what PySB is really doing is kind of taking those things and grouping them in a functional format to relate them to the experimental data, so we don't lose the resolution across the way. So what we gain with this is that we can write models in a common language. We can extend it beyond simple rules. It's open source. We can leverage modeling tools that are existing out there. For example, PySB right now talks to BioNetGen, to Kappa, SciPy, and NumPy. We can leave the mathematical details under the hood when it's needed, or we can explore them when we want. And importantly, we can facilitate model extension, exploration, sharing, and I have access to the open Python environment, which as of yesterday had 78,000 packages on the repository. So this is just a plug for our tool. So I'll tell you a little bit of what we've done with this. So we took this model. So what we did, we said, well, let's look at this proliferation of this cancer cell proliferation work, and we decided to just build a model to represent the different clones. And we start with something very, very simple. Of course, our eventual goal is to link the signaling to the cell behavior, but we start with something very simple where we just said, a cell I can either divide and make two cells, and the daughters inherit the whatever rate they have from the mother cell, or they can die. And this is just how we probe these things. So we make this model where we say that okay, the derivative of the log of the cell growth versus time, or the number of cells versus time, is just the difference between division and death, and this actually relates directly to the experimental dip rate, or the drug-induced proliferation rate. So we can actually relate this to the experiment, and this is in contrast with with the sort of deterministic view that people have come up with. So in this again very simple model, each cell type has a unique division rate and a death rate. The rates are the cell types inherited by the daughter cells, and there's no transitions between cell types, at least initially. And this is actually a key point that I'll point out later, because this will come out to be important later. So, now we have the model. We've encoded it into PISB, and the question is, well, can we sample this proliferation and death rates from the experimental data? And the idea was, well, if we do deterministically, we're going to get this, so this is not very interesting. So once we have the model in Python, we can just explore it in many other different ways. I don't need to introduce everyone here to this, but I'll just go very quickly through this. We use the Gillespie algorithm, or the SSA algorithm, to analyze the system. And what we, as you know, in the limit of infinite copy numbers, the solution approaches the ODE deterministic solution. And in the limit of an infinite number of simulations, this provides an exact solution to the chemical master equation. And this is work by Dan Gillespie from 1976. And this is just an example of where this at a low number of copies, there's a lot of noise. The dashed lines are the ODE answers. And as you increase the number of copies, you approach and you do many simulations, you approach the average solution. So our idea was very simple. Can we just use this model and just use cancer clones as some Frank coins? So can we actually just say, some cancer clones have different chances of responding differently, and can we actually just sample from this distribution and look at how the cancer cells proliferate? So we did that, and again we go back to the experimental data, and we sampled the division rates from these distributions. No, sorry, this is the growth rate distributions. And what we did when we sampled this from experimental data is that, remember, the experimental data has a lot of averages. What we saw that we could capture the noise, but the noise was not very much. So we started to think, well, the reason why this is like this is because of this difference between the division and death rates. So there must be something that we're not capturing here with the noise. Is it the signaling? Is it whatever? So I told Leonard to, and by the way, this is again PISB calling BioNetGen and calling the Gillespie direct method from Stokit. So I told Leonard, how about we start playing with the parameters and seeing if there's something about this division differences that we can actually play with. And when he did that, he started to play around, and he saw something that was kind of interesting. If he left the cell populations that would supposedly disappear eventually because they have a negative dip rate at this low division death difference, and he increased. So this is now from 0.02 to 0.2. He increased the division rate in the ones that go faster, in the ones that eventually grow. He was actually able to recapitulate the experimental data. And this was very exciting, because what this is saying is that if this is true, we should be able to look at the experiments and observe some cells that are some cell sublines or subtypes within the clones that are slow dividers and fast dividers, and they should behave differently in terms of drug exposure. So this is just N52 simulations here to show you an example, but we did thousands of simulations, again running this using these three tools, and in PISB this is just a simple for loop. So this explained several things. So first of all, we're able to recapitulate the experimental data just by adding noise, which was kind of neat. And then the other thing that was interesting is that in the experiments they often saw that some cells would would respond dramatically to the drug, even if they were in the clones that had negative dip rate. And some cells were very poor responders, even if they were in the clones that were very sensitive to the drug. And we see the same behavior in our simulations, where we have fast and slow rebounders. For example, here we have this population that went down a little bit and went back up really fast. This population that just was very slow. And similarly, we have even through population different dip rate in the population. We see examples where some cells some cell populations just kind of stay stable, or for example this one, which is supposed to be a clone from a negative dip rate, just by tossing the coins randomly, somehow just responds very dramatically to the drug, or something with a positive dip rate that should just continue to grow, just by tossing coins, eventually exhibits this behavior where it just goes down. So we're seeing that there's a component of noise in these things that we don't understand, and what Eric talked about in the previous talk, this heterogeneity and how the cells are managing this information processing is not straightforward. So we did this. We wanted to make sure that we were doing this with doing something with that was generalizable to other drugs or to other cells, and so we did this with different cancer cell lines. So this is lung cancer. This is I think this is carcinoma. This is and then we treated with different drugs, erlotinib and TNF, and ABT, which is erlotinib and ABT, which are different drugs. And we were again able to recapitulate a lot of this noisy behavior in the systems. So the question was, well, what is it that we're doing here? How are we understanding these systems? So right. So just to kind of summarize at this point. So what we've done so far is we've said, okay, there's these cells that are oncogene addicted. They're carcinogenic, and they have different displayed resistance to the drugs. And what we see is that just for some intrinsic noise or some way of noise, they can display different responses to the drugs. And the question is, well, what is the origin of that? And that's where we're going next. But we see that in the populations, in the whole population, there's some subpopulations, and I'm drawing this as to just illustrate that there might be many different minima in this thing. There's some populations that are very sensitive to the drug, or sorry, some very resistant to the drug, and some populations that are very sensitive to the drug. And not only that, we from our clones, from our cell populations, we can see that different seedings of these different division and death rates can give different behaviors. So so the question is, is it that there's different substates where the cells can jump and can move around? And this is something that people have shown. And certainly, if we take from our data that the different ratios of division and death are somehow representative of these different states, then that's certainly something that that our data might actually favor. So so the question next to us, well, can we do better? So we're doing again, like I said, this was very preliminary. What we have done now is we've actually gone to the experiments, and we do find that there's the fast dividers and there's some cells that are very sensitive to division or death, and they make this decision very quickly. And then there's some cells that are actually very tolerant to the drug, and sometimes they seem to die, but then eventually they transfer to this kind of a tolerant state that we hypothesize could eventually develop into a resistant state. So what we're doing to improve these models is, well, we're basically doing two things. The first thing we're doing is, in my lab we worry a lot about model calibration, how models relate to experimental data. And what we're doing is, in this case, we're using this Bayesian model calibration, which is called DREAM. It's an adaptive Monte Carlo method that one of the difficult things when you do Bayesian inference methods is that the sampling is very difficult, and it takes a long time to do these things. But this one is parallelizable. We published this in PNAS last year, and we're going to publish the actual method very soon. But the idea is that even if you cannot measure everything in the system, if we're going to start, especially if we're going to start adding transition states in the system, the question is, well, how do the parameters relate to the data? And even though we cannot measure everything, we can start to say things, well, this parameter is very constrained given to your relative output. So those parameters you should focus on a lot more, or your parameters are very broad and your system is very insensitive to those parameters, and you don't have to worry about that so much. So that's one of the things that we're kind of aiming forward. The other thing that we're trying to do is, and this relates a little bit to what Omer talked about in the first talk, is model selection. Many times so as I said, this model is very simple, but there could be multiple transitions, and the question is, well, how do we actually relate those models to the experimental data? And what we have done is we're trying to use optimal scaling in combination with machine learning. In this case, we're using TensorFlow, which is the Google machine learning tool, to basically take experimental data that has very little quantitation or no quantitation, things that are ordinal or just have different things that relate to experiments but are not necessarily numbers, and do this calculate this likelihood functions and look and see if we can extract better parameterization for these models. So this is again work that's ongoing. In this particular example, we're looking at complex one formation from TNF ligand signaling. This is, we test 14 initial conditions, 67 parameters, about 700 equations. This is something that is it can be pretty daunting, but with this type of methods, we can actually extract parameters and start to tell you, focus here, don't focus here, and this is how we can sift through the noise. So, so this is where we are. I'd love, if you guys have some input or some ideas, I'd love to hear from that. Just to again, send this message home, what we've done with PySB is we have a Python-based tool that can link, simulation, visualization, calibration, modeling, and analysis, or within an open source framework. We can, for the simulation side, you can use, stochastic simulations, Gillespie. We're trying to connect now to PyRDME to have stochastic and spatial. We even have a tool that connects to Smolden if you want to go fully particle-based. So once you have your model built, you can simulate it in many ways. There's lots of tools for visualization. We're connecting to tools that do different calibrations, including our own, and more importantly, we connect to different modeling tools in the field, like Kappa by NetGen, and so on. And there's tons of analysis tools. So this could be, if you're interested in exploring this more, I'm happy to tell you more about this. So so I'll just I think I'll end it here, and I'd rather have more discussion because this is kind of this is just our simple message today. So just in summary, we have the PySB tool for model building. We can use multiple simulation methods once the model is built. We show our role in this very simple system for noise in cells in response to drug perturbations. We predict fast and slow responders in the cells that we have now. I didn't have this data, but we have now verified this experimentally. And the exciting thing is that these cells seem to be, even though they're genetically equivalent, they seem to be in very different signaling substates, or we think it's related to transcription factor networks. And we see that even though this intrinsic stochasticity of birth and death events explains aspects of variability in drug response that we're now trying to look more into in terms of transitions across different substates. And then finally, connecting this calibration and model selection to experimental data, especially with stochastic models. So I'll leave it at that, I guess. It's a bit early, but we'll talk more. And this is just my lab. Aaron did the work on the calibration. Michael did the work on or is doing the work in the machine learning, and Leonard did most of the work for the stochastic cell simulations. And this is our funding sources. I want to thank the organizers, David Anderson and Sam Isaacson, and our collaborators, and I'd be very happy to have some discussion.
Thank you very much for your talk. We have plenty of time for comments.
Yes.
So the heterogeneous responses of apparently clonal tumor cells is a really interesting phenomenon. Has there been any? I hesitate to ask this at this conference. Have you done any single-cell RNA-seq type of experiments?
That's a great question. So there have been experiments of people doing not single-cell. Oh, no.
If you can, even if you can isolate clonal populations derived from a single cell, that would also probably be.
So there's a work by a group in Harvard, Engelman, that has done something like that, but they haven't done it at the single cell level, but they have separated the clones, the subclones that respond differently to drugs, and they have seen that they respond differently in different ways, and looked at the gene expression or the RNA-seq level, and they see differences. They see actual differences but they cannot correlate that directly to the actual response data. That being said, we're actually trying to do nanostring experiments in our campus, but they're not as trivial as you think, you know.
I mean, how certain are you that they're actual genetic clones? So I mean, the method is you're starting with a clonal population, and there must be some correlation time for which they have nearly identical phenotypes, right?
That's a great question. Yes. So from the work by where's John when you need him? From the work by Peter Sorger and Sabrina Spencer, they showed that that the correlation for response, at least in HeLa cells, was about 2 divisions. In our case, we're actually seeding the cells from the same single clones and building colonies, and we let them go for about 10 or so divisions, less than 40, and we certainly in that time we don't expect any major introduction of genetic mutations. We have tested we've done ChIP-seq experiments to test for potential mutations in these clones. And we know for sure that there's no, for example, P740M, which are the traditional, EGFR mutations that make the cells resistant to erlotinib. The other thing we've done is, the name escapes me right now, but we've used chromatin assays to see how accessible the DNA is in the different clones. And we see that there's actually some variation from one to the other, despite the fact that they're mostly genetically identical. I will say that we haven't done any deep DNA sequencing, so I haven't gone that far.
From a more sort of theoretical perspective, I wonder if you could show that there's some kind of multi-stability phenomenon going on like that, the Waddington landscape type of.
Right. So that's kind of what. Here, let me fast forward really quick. So absolutely, and this is actually what we're aiming for with this view. So we do think that there could be a sort of Waddington landscape thing where the cells are jumping from one to the other.
Sorry, I was not paying close.
No, that's fine. But it's good because the possibilities are the problem is that if we do this, then we end up with something like what Omer did. Where's Omer? Omer did, where you have to look at thousands of connections and how many millions of parameters, and then you end up with a big mess, and it's very hard to tell what's going on. So what we're trying to do is actually use statistical tools to actually extract this and calculate. In fact, I wrote it here, the probability of a given model being true given a set of data, or the other way around, and you can do that from a Bayesian perspective, and then come up with the top 2 or 3, kind of Waddington landscape views that will give us the same results with the same data. Yeah, go ahead, Omar.
So I'm just kind of curious, is there any way to determine a timescale for the meandering between the sort of...
So the hard thing is that, so theoretically we should be able to, and we should be able to characterize that in terms of the time that it takes for the cells to divide. From what we've seen in other experiments, things like chromatin rearrangements they can take 2 to 4, 2 to 5 divisions or something like that. So we would expect it's actually heritable, but it's not genetic. So it's very interesting because the sister cells, once they divide, they could be in one of these landscapes and jump to another one over 2 or 3 divisions, and then they could go their own separate ways after 5. This is something that we're gearing up to test. It should be possible to do things like histone, characterization or chromatin, characterization to do this. But most of those are destructive experiments where you have to kill the cells.
What if you just let them, sort of continue to proliferate in the presence of drug?
So if you do that, they become resistant.
mutations as well. Yeah.
And eventually mutations will set in. We have tried taking the cells, removing them for drug, and letting them grow for a while, and then putting them back in drug, and we see the same behaviors. At least in under 40 divisions or so. Once we get past 40 divisions, then we cannot rule out mutations, and it becomes a little bit more complicated. We have tested again for the most common mutations in this. So this is just to go into the details. Erlotinib inhibits the EGFR pathway, and then there's some magic crosstalk that people still don't understand, and this engages the apoptosis pathway, and then cells die, like they showed us earlier. But we know that if we inhibit the EGFR pathway, most of the cells die, but somehow there's some mutations like RAS mutations, RAF mutations, and EGFR mutations make them resistant to the receptor. And after definitely over 100 mutations or so, you'll start to see, 100 divisions or so, you'll start to see mutations. Yeah. Yes?
This is a question more of clarification. So if you go back to those plots where you have things going down, you have a whole bunch of trajectories, and there's quite like these things.
Yeah.
So I guess that the point here when you're doing these simulations is that each one of those trajectories starts off with a single cell that in this case has an individual growth or difference that's drawn from that distribution
Yes.
it started with.
Yes.
I see. So this is essentially saying that if we were to do an experiment where we take those clonal populations, each of which has this different average difference in division and stuff, and we then sample from that based on the empirical features of that distribution, can we then replicate experimental trajectories of the cells dividing individual sort of cells from each of those clones?
That's exactly right. That's exactly right. And what we're saying also is that, so what this experiment does effectively is, if we believe this picture, of the different, substates, what we're doing is we're taking cells from one of these wells and just growing them independently, and we're trying to look at their behavior and response to the drug independently, and then putting it back together numerically.
So have you looked at what amount of this variation comes from the initial variation in those states, say, versus, because you were sort of saying something about how you obviously have a stochastic model of the cell growth process as well. You've got a single starting cell. So which of those things dominates in driving the variance? The variance of the underlying difference in rates, or the variance
what we
what
we find is that the division rates, the fast dividers, seem to be the drivers of the variance. They're the ones that seem to.
So you have to make the division rate of the fast guys in this distribution high in order to replicate the variance that you
Absolutely. And this is where I think it's interesting, because then the question is, well, what does that mean in terms of the signaling? Because that means that information is being processed differently by the fast dividers versus the slow dividers. So that's that.
Any more questions or comments?
Yes, go ahead.
It's the machine learning aspect.
Yes.
So you're not actually sort of building a model, say, based around an ODE. You're doing some sort of polynomial chaos expansion.
We are doing
Basis functions. Is that how that works?
So I remember that. Okay, this is where I'm not the super mathematician I claim to be here. So what we're doing is we are just we're doing a non-ordinal kind of chaos expansion and letting the system pick the terms that matter most to the.
Some expansion within polynomials of certain degrees.
Exactly.
Or whatever.
Yes. But we're doing it actually with Gaussian seeding. Yeah, and what we're doing, for example, here is that when you have multiple data sets that are overlapping with each other, we're trying to learn from the machine. If you're here in this distribution, how much of this goes into this classification versus that classification, and we're letting the algorithm do that for us. And that's actually the hard part, being able to tell, well, of this information, how much belongs in this bin versus that bin.
And do you find that algorithm, does it scale particularly well?
I'm sorry?
Does it scale particularly well?
It scales pretty well, actually. So we started out with scikit, which is the one that everyone uses in Python, and that worked. That didn't scale very well because it doesn't handle ordinal nonlinear classifiers very well. But this TensorFlow tool from Google actually works really well, and it's very parallelizable, which is really nice. So this is not published yet, of course, but I'm happy to share what we have if you're interested. All right. No, she just was raising her hand too. All right. Thank you very much, then.
No questions. That's fine.